in the room, could you put your hand up? $11, okay. Yeah, great. Thanks, everyone. And then in regards to the hikes, there will be some information uploaded to the website, thanks to Jeff's wizardry. Thanks, Jeff. So check the website in an hour or so, yeah, and we'll be good. Anyway, it's a pleasure to have Andriy Marusyk here, who's been at Moffitt now since 2015, so coming up on 8 years. And Andriy is really, he thinks like a modeler, I always say to him, and he really works great with modelers. And if you collaborate with biologists or with oncologists, you know that there's many of them who don't fully grasp what it is that we do. And then there's some that really embrace it and understand it in a way that helps you even understand your work in a different way. And Andriy is definitely one of those people. So it's cool to have him here to tell us all about the role of fibroblasts in tumor growth and treatment response. So please take it away, Andriy.
Yeah. Thank you, Sandy, for your very nice introduction. I've never done this kind of didactic lecture before, so had to make it from the scratch, and I hope it would make sense. I try to make it useful. I'm not sure about the timing. I'll try to keep on time, and then if you see something that sounds interesting but not quite clear, please feel free to talk to me afterwards. So again, I'm gonna spend about half of the talk trying to introduce the key ideas and concepts that might be helpful in thinking how to approach modeling cancers and think about cancers. And the second half of the talk, I'll introduce some of the lessons and inferences that we've drawn from the studies in the lab. So my training was in experimental cancer biology, which was and still is dominated by fairly reductionistic molecular oncology paradigms, where you classify the cancer phenotypes based on the hallmarks and try to identify genes and pathways that controlled by these genes with the purpose of developing therapies that could suppress this hallmark and cure the cancers. So obviously there are many success stories. Most of the targeted therapies we have on the market are a result of this reductionistic approaches, but it's also becoming obvious that this approach has limiting returns, and we are getting longer and longer list of critical regulators of specific hallmarks and specific cancers, and it's not quite clear what this knowledge really gives us. So I think we need some organizing principles. And there's also a lot in the blind zones of the mainstream efforts that I think requires modeling to make sense of. So as an example, I'd like to show this tumor model that we described in the paper that I've written with Philip Altrock during postdoc studies. So the weird thing about this MDF 468 tumors, xenograft tumors growing in the immune compromised mice is that their growth is very, very slow compared to some other models. Well, you can be fit into exponential tumor growth, but the question is, okay, why does this tumor grow so slowly? And a reasonable guess is that, well, maybe the tumor cells are not proliferating much, but that's not the case. If you look at the markers of proliferation, the cells within these tumors proliferating a lot. So the question is like, okay, how come that? And in fact, like if you compare the proliferation rates between the two models, they're nearly identical. So what's going on? Why is one tumor growing and another one is not growing? At that point, I concluded that, oh, the death rate must be similar to birth rate. But this is kind of a trivial conclusion. Of course, like if the population is growing, the birth rate must be higher than the death rate. If it's shrinking, the other way around. This thing means stability. And if you look at most of the natural populations, if it's stable, it means that the birth rate is similar than the death rates. But like, what prevents this tumor from growing to the same extent as the other one? And in this case, it was the case of having hard time pushing the butters, which we can revert by introducing specific mediators of the crosstalk with microenvironment. But another thing that I wanted to point out, because I've seen quite a few examples of this in high profile papers, even in Nature. Okay, so the death rate must be similar to birth rate. One can throw the markers of the cell death, in this case, tunnel staining or cleave caspase 3, and you typically see very few dying cells. And in some cases, in way too many cases, people conclude, oh, that the death rates in this system must be insignificant. But this doesn't make sense because the tumors are quite stable. But what it means is that, well, the temporal dynamics of being able to detect the marker of death is minutes versus being able to detect Ki-67 is many hours. So my point is, in order to make sense of things like that, obviously there's this hallmark paradigm, and drivers and passengers do not really give the answers. What we need is to have some sort of a quantitative reasoning to make sense of what's going on and how things are changing in the space and time. And in addition to that, we need some organizing principles to think about tumors. And there are, I would say, like two main ideas to think about cancers. And I think they are both incomplete by themselves. It's almost like particle and wave duality in thinking about light. Is light particle or is it wave? You have to think about, you have to consider both to make sense of what's going on. So the one paradigm that most of you are probably would agree and know much more than average cancer biologist is that paradigm of cancers as evolving ecosystems where neoplastic populations evolve due to the diversification of heritable and semi-heritable phenotypes that enable them to adapt to changing conditions and things like metastatic niche or treatments. But at the same time, the cancers are represent the case of messed up differentiation and tissue regeneration processes. And I don't think one can reduce it entirely to an ecoevolutionary paradigm. And also, let me give you an illustration of that. So this is a teratoma, fairly rare tumor that occurs during the embryonal development, where you can have this ugly, aggressively growing tumors, but these tumors are quite different from most of others in that they have a kind of caricature of organ development. You can even have teeth, and they're much more uglier picture if you want to Google. But the point is that, like, it's hard to make sense, like what's the sort of fitness function, etc. It looks like it's some confused stem cells started to try to make the normal tissues and stuff in the inappropriate context, resulting in this bizarre thing. One can also say that, okay, these tumors are fairly rare. Let's ignore that and focus on something that's more common. But there's a kind of an opposite example. In this case, this is a work of Jill Smith lab from NIH, who took metastatic human breast cancer cell line. And if you inject these cells into fat pad of the mice, they will form tumors. But if you mix these tumor cells with excess of normal epithelial progenitors, they can become part of normal epithelial tissue. So they behave as normal cells. They can even produce milk. So again, like in this case, like how do you make sense of a tumor cell that suddenly starts behaving like a normal cell? It's almost like some gang member being transferred into a normal neighborhood, and with good neighbors, he or she behaves more reasonably, maybe with higher chance of relapse. And then there's a third observation. It's unpublished in my lab. I'm still trying to wrap my brain about it. So in this case, we were doing tail vein assays to look at the metastatic growth in lungs. And at that experiment, we didn't have enough female mice. It's models of breast cancer, although one can say that the male breast cancer is also the thing. So we took male recipients, and some of the mice developed tumors as kind of the way they would expect them to look. but a subset of tumors were very bizarre. To me, it looked like a picture book of a histology of the testis tissue, and I brought it to our pathology and asked, like, what is it like? Is it, of course, what we're asking is testis. Haven't you had your biology courses? And yeah, this is kind of what the testis tissue looks like. And we haven't done the staining, but looks like inside of these things, there might be even like a semen tissue. So the point, again, these are kind of weird things and outliers, but I think they kind of illustrate the point that that we need to think about thinking about cancer. We have to think about them as tissues and have to consider the developmental paradigm while making sense of what's going on. So again, like I think that to make sense of cancers, you have to treat them as tissues. Just wanted to give a very brief intro of what the tissues look like. Of course, different tissues are quite different. The majority of cancers in humans are of epithelial origin, carcinoma. So therefore, I'm going to show an example of organization of epithelial tissues. And of course, devil's in the details. A lot of them have very important details, but there's still similar kind of generalizable organizing principles. In this case, it's breast epithelia where you have the duct, the branching epithelial tree that is located and surrounded by stroma. So the breast epithelial, you have two layers of cells, myoepithelial cells and the luminal cells. The luminal cells are the ones that make milk, and the epithelial cells are separated from stroma by the basal membrane. Importantly, the epithelial tissues are two-dimensional. So even if a cell that has a potential oncogenic mutation, its ability to expand is fairly limited. So maybe let's see if I can use this. So just here just an example of how things look at the staining. Here you see kind of a lumen and ducts. So lumens are the major place where milk is made, and the ducts move the milk to the nipples. And so different epithelial tissues are made differently, but the general principles of organizations are similar. And so what's about the stroma? So the stroma in breast epithelium is fairly cell poor. You have fibroblasts, you have immune cells, you have vessels, and you have lymphatic vasculature. So it's essentially providing a infrastructure and support for the main sort of functional part, the epithelia. But it's also could be fairly dynamic system where, like in particular in breast with the estrous cycles and during pregnancy, there's dramatic remodeling in this epithelial tree. And so for this remodeling to proceed, it depends on like highly kind of controlled, choreographed interactions between the different types of cells within stroma and epithelia. So what's going on with cancers? And the best way I think to grab the ideas is to take advantage of the power of metaphor. And I think there are two strong metaphors that I'm finding extremely useful. One is that of tumors as caricatures of normal tissues. And obviously the teratoma is a perfect example of a caricature of normal differentiation. But in most cancers, like in breast cancers, it's not so obvious. So I'd like to unpack it a little bit. So what are the features of the caricature? So one thing, it has to represent the kind of the original, but in some messed up and hypertrophied form. So in cancers, in epithelial cancers, you still see the same tissue components like in normal tissues, but your epithelial compartment goes like way out of the whack. Your stroma is different. You have infiltration of the immune cells that you don't see in the tissues. Another kind of beauty of the parallel of caricature is that you can have multiple caricatures of the same person that have portrayed somewhat different aspects of it. And yes, so different tumors have different evolutionary trajectory. There no tumors are alike, but so they can represent fairly different messed up versions of the normal tissues. Another good metaphor is the tumors as wounds that never heal. And One experiment that come in mind is the classical study from Mina Bissell lab that was kind of violently rejected by some subset of cancer biology field. But the point here is that this is like one of the first oncogenes that was thought to be both necessary and sufficient to cause cancers. And what Bina's lab demonstrated is that if they introduce VSARC, this powerful oncogene, into an in ovo, in the egg, the chicken that develops from this egg look normal. They don't have tumors. They do have cells that express VSARC, but they look normal. They don't have any hypertrophy. But if you introduce, if you scratch the tissue that have this cancerous cells, they very quickly develop these tumors. It looks like almost like a kind of state transition. And there are sort of somewhat similar example, though it's, things are much more murky in human patients because obviously you don't do things experimentally. But let me unpack this metaphor of wounds that never heal. And to do that, let's sort of think about the wound healing process in normal tissues. And since all of us, I assume, had cuts and bruises, it's more intuitive probably. So what happens if you cut your skin? You are damaging the epithelial layer, you're damaging the basal membrane, and it leads to activation of residual tissue fibroblasts. It leads to other sort of both fibroblasts and the dying tumors, dying epithelial cells induce the clotting response. It induce migration of the macrophages and other immune cells that take care of the bugs that got into the wound. You can get angiogenesis, you have swelling, you have redness, you have more blood flow to the tissues. And normally what happens is that this acute phase of the response gets the tissue, the damage resolves, the layer, the wound gets repaired, and you return back to normal homeostasis, maybe with a few scars. But what happens in cancer is essentially initially the same sequence of event. Instead of a cutting wound, you have abnormally proliferating cells that keep proliferating and dying, and they're releasing the same damage signal as the wound, the cutting does. And it also leads to activation of the tissue stroma, the angiogenesis, but in this case, the wound is never healed. And so the cycle gets repeated in perpetuity, and the resulting tumor cells are evolving and changing. So, and Sandy mentioned the fibroblasts in the introduction. So fibroblasts are the major player responsible for the stroma. Of course, it's all the network of interaction between different cells, but the fibroblast is the most kind of plastic cell type that can differentiate into pericytes, it can differentiate into adipose tissue. The fibroblasts are producing the majority of extracellular matrix that connective tissue is made of. They secrete a bunch of things that mediate communication with different cell types, and they can be active participants in the immune response. And wanted to kind of give some examples from our studies. So in this case, in the middle, you have a picture of tumor cells plated in metrogel, and originally they formed this kind of small clusters. But if you co-introduce fibroblasts, what fibroblasts are doing, they're making this ECM glue that helps stick the cells together. And they have the contractile function that bring cells and organize them in larger organoids. Another thing that was like very surprising to me at that point, we had just simply co-culture between tumor cells and fibroblasts. And if we're looking at the changes that are induced in tumor cells in co-culture with fibroblasts, and there are no other cell types, and the half of the changes are related to the response of tumor cells to the immune system. So it looks like the fibroblasts influence the tumor cells in a way that primes their response to the immune cells. And the different cell types have variable responses, but you could see this pattern that's like applicable across the board. Sorry, wrong one. Another very important thing is that if you look at the consequences of in vitro cultures of fibroblasts and tumor cells, another striking thing that jumps out is the induction of EMT, epithelial to mesenchymal transition, where the epithelial cells partially dedifferentiate and acquire migratory property. So in here you have just fibroblasts themselves or tumor cells by themselves. And in co-culture, some of the tumor cells start to look indistinguishable from the fibroblasts. And we see the same thing in the tumor tissue where in this case, we don't have specific staining for the stroma, but it's rather kind of cell poor dark areas where where the fibroblasts are seen as basically this elongated cells without fluorescent staining, and the tumor cells are rather red and green. And you could notice that the cells that are adjacent to stroma have acquired the same elongate shape of the nuclei as fibroblasts, whereas the cells that are farther out retain their kind of more normal shape. And why it matters? Because it's really relates to most dangerous properties of cancer: one, ability to migrate, move, and then the ability to change the phenotypic state through state transition, to essentially ability to adapt through phenotypic plasticity. So it makes sense in normal wound repair, because if you cut the skin, you have normal cells that are capable of dedifferentiating, migrating to the wound site, and then dedifferentiating. And the problem, of course, is that in cancers, the dedifferentiation does not necessarily happen, and you can have this vicious cycle, plus the ability to invade, plus the ability to adapt to new stressors through epigenetic mechanisms. So, and one more additional thing I wanted to go through, kind of the interplay between tumor cells and stroma during the cancer initiation and development. So it's all about the interaction between this tumor cells, epithelial cells and stroma. And there have been studies that show that the genetic changes, for example, PA3K mutation, no, actually it's a PTEN deletion in fibroblasts can actually lead to dramatic increase in, is capable of inducing carcinoma in mouse models. So, but normally what's happening is that the cells, as long as the cells are remaining in, sorry, I forgot this is not a laser pointer. As long as the cells remain in 2D, they're more or less okay and under control. They're basically surrounded from all the sides. I think what is extremely important is essentially a niche partitioning event when the cells acquire capability of going from 2-dimension to 3-dimension. So tumors are 3-dimensional structure, epithelial layers are 2-dimensional structure. So the cells acquire the access to new resource space, but then of course, like they lose the access to normal nutrients supply to the oxygen supply, and it can induce the cell death, but then it can also lead to this cell turnover that leads to release of wound signaling, of death signaling. But as long as these things remain in the duct, this is considered to be a benign disease. And in some cases, especially when there's not much of a turnover, these are fairly low risk. But unfortunately, a subset of this, and especially this is characterized by excessive necrosis and cell death and turnover, the basal membrane can rupture, and this enables a direct contact between tumor cells and stroma, which really instigates and starts this wound healing cycle. So next, I would like to show some of the things that we've learned from our experimental studies in the lab. And we're working with different models, but all of the data that I'm going to show today was done with experimental models of lung cancers. And we are focusing on ALK-positive lung cancer, which represents a poster child of both initial success of targeted therapies that can induce very strong and powerful remissions. And in some cases, these can last for years. But unfortunately, inevitably in patients with advanced metastatic cancers, these the resistance is unavoidable, and the tumors will eventually relapse. And we have experimental model both in vitro and in vivo that recapitulate this response with different temporal kinetics. So what we're trying to do in the lab is to understand how the resistance in the initially sensitive tumor cells populations evolve. So rather than starting with the favorite assumptions, we're trying to be as open-minded as possible in figuring out what's going on, and then capturing this understanding in the mathematical models and testing these models by formulating predictions that can be for experimental observations. That it essentially enables us to play the Groundhog Day scenario, where you can, if you get unsatisfactory results, you can go back and try to refine until you get things right. And I think with this paradigm, we can make sure that, of course, like no model is perfect, but as we can make sure that the models are good enough to help us guide the development of evolutionary informed therapy strategies. So this is more kind of goal rather than the reality at this point, but I think we are moving in this direction. And before I show some of the data related to tumor-stroma interactions, I think it's important to show some of the kind of key piece of evidence on which a lot of our current thinking is based. So there's lots of talks yesterday and today talking about the adaptive therapy, the idea that before you start the treatment, there is a subpopulation of resistant cells that have a fitness disadvantage, and there are in low numbers. And then as you start the treatment, you kill off sensitive cells, at least you get competitive release of these cells, and that's how you get resistance. And we initially thought that we would get a similar picture, and instead we observed something quite different, where we hit the cells with a drug. We can only see cells that are barely surviving. This is not particularly new. It's known as therapy persistence, resistance, or tolerance. It seems to be fairly widespread across different cancers. There's good evidence that this is what's happening in many patients. But the thing is that these cells acquire resistance not through a single mechanism, but through a number of epigenetic and genetic changes that are happening in the same cells. So the path to resistance is not a binary switch, but instead appears to be a trajectory both in the adaptive landscape space and in the epigenetic landscape space. So these studies were mostly in vitro, but we did experiments where we taken resistant cells and mix them with sensitive cells, or rather naive cells, because these cells can develop resistance. And we subject the mice either to vehicle control or treatment. In the absence of the ALK inhibitors, we don't see an obvious fitness defect. We get the proportion of cells remains the same as we injected. Upon the treatment, however, we get a fairly quick expansion of resistant cells that go from 1% to 50%. And we're seeing an almost immediate relapse that starts happening after a week of the treatment. So if you have the scenario in clinic, it wouldn't be even detected as acquired resistance. It would be considered as intrinsic resistance because of the spacing between the tumor scans. So again, like, I'm sorry, I'm using the wrong thing. So the implication is that when you're facing the situation like that and you start to model it as if we have a pre-existing resistance, no matter how great mathematician you are, if the assumptions are incorrect, the conclusions of the modeling could be quite misleading as well. And actually talking to some of the clinicians and experimentalists, people say, okay, well, yeah, it's nothing new, of course, like everyone knows it, but well, why everyone is publishing this with a paradigm of the single resistance mechanism? And then I think it often reflects just the pressures of publications and grants. So if your gene is one of many critical regulators of resistance or persisters, how do you sell your idea to a granting agency or to the journal? But in order to succeed, we cannot pretend that the reality is much simpler than it really is. And maybe there are contexts where we can ignore these complexities, but I think we have at least have a kind of adequate understanding of what we're dealing with and what their other rules of the game. But getting back to the issue of stroma, how that gets into play. So in the previous data, we mostly had in vitro studies. We know that contacts in vivo are quite different. They have a different replication death dynamics. You have gradients of oxygen, nutrients, pH. And there's another very important point, and actually our Bob Gatenby was like one of the early visionaries who kind of co-authored that paper that pointed out to the observation, which is now fairly well accepted in the field, is that if you co-culture tumor cells with fibroblasts or their conditioned media, you can get a remarkable protection against most types of small molecule-based targeted therapies. And so how do we deal with it? And there are essentially kind of two main approaches to deal with it. People either tend to completely ignore the possibility of its existence, or they're trying to explain relapse based on these effects. But it doesn't quite make sense because these signals are acting over short distance. So in principle, only tumor cells that are close enough to the stroma might be protected. So we decided to look at it directly. And in this case, working with animal models, we can sample tumors at and having a fairly predictable responses enables us to sample the tumors at different times at the remission and relapse growth curve. And what we're seeing here at the baseline, we are staining here against the marker of proliferative cells. This is BRDU. It captures the cells that are going through the active replication of DNA. At the baseline, the distribution of cells appears to be fairly random. As soon as 2 days post the treatment, you start seeing this fairly obvious patterns forms of proliferation. And these patterns actually follow the patterns of stroma. At 2 weeks, there's clear increase in the stroma, and stroma in this case is sort of dark blue. This is just counterstain of immunohistochemistry. And you're seeing that you started that the proliferating cells, we see them preferentially or almost exclusively in very close proximity to stroma. So even at point of the relapse, where the tumor cells now can survive and grow outside of the stromal niches, there's still some remaining association that persists. And so what I hope you can appreciate from this picture is that there is a dynamic process where both proliferation, tumor cells, death of tumor cells, the stroma, their shape and the amount changes over the course of the treatment. And so we're trying to kind of make sense of, I think we need to make sense of what's going on in order to adequately understand the process of acquisition of resistance. And just looking at the slides could be useful for getting some sense, but it requires, again, like quantitative reasoning to get a deeper understanding. For example, looking at this picture, it's not super obvious whether the patterns of proliferation are indeed random or not quite. Maybe there is some bias and we're good in capturing bias, and sometimes we're capturing bias where there's non-existing. Or if you look at this and this one, it's unclear whether the association between tumor cells and stroma is stronger in one versus another. So to deal with this thing, and luckily Tiana Miti, a very talented postdoc that we're commenting with David Basante, is giving a talk tomorrow. So she will talk about the technical details, but she worked with another postdoc from my lab, Andriy Marusyk, who figured out the reliable segmentation. So essentially we developed a digital pathology pipeline that enables us to apply spatial statistics metrics to look at the strength of this association between stroma and tumors, contrasting it to the scenario of neutral and random distribution, as well as the distance of the signaling. And it looks like from this analysis, it appears that at 2 weeks, the effect is the strongest, and it goes down over time, but even at the relapse, we see essentially the same strength of association as in 2 weeks of the treatment. So based on this data, it appears that the cells that are close to stroma are the ones that are capable of surviving and maintaining their proliferation And over the course of the treatment, the cells must somehow figure out how to acquire the ability to survive outside of the stromal niche. And this is what drives the relapse, at least in this experimental system. And another very important point here is that when we look at the spatial transcriptomic analysis, we're trying to assess what's different between cells that are close to fibroblasts and those that are far from the stroma. And we're seeing, again, like evidence of very strong EMT signature, which is associated with plasticity. So it looks like the cells that are close to fibroblasts, they're not just surviving, but they're also might have an enhanced ability to change phenotypically and adapt to the stress of the drug by rewiring their gene regulatory networks. So this quantitative knowledge then enables Tatiana to start building the in silico ABM models where we capture proliferation death dynamics as well as the impact of the stroma, both the strength of the impact and the distance of the impact. And if I still have a few minutes, I wanted to highlight another thing, getting back to that slide that I showed with the about the pre-existent resistance. So in that experiment, we use about 1 to 500 ratio, and one can say, well, the human tumors are much larger. There are many more tumor cells, so that this like models don't, and you can have a much lower frequency of pre-existing resistance. What if it's just only one cell? You cannot model it directly experimentally. Even if you put it resistant cells at the implantation, there's no guarantee it will survive. And by the time the tumor forms, you might have thousands of copies of the cells. So instead, what we decided to do is we decided to make essentially a series of limiting dilutions, getting progressively fewer and fewer tumor cells. And we're looking at the expansion dynamics. And the idea was to use the power of mathematical modeling done with very two talented at that point high school students who are now college students under the mentorship of Mark and Sandy. And so the idea was just to figure out the limiting case of where you have only kind of one or a few tumor cells. And once we tried to do that and predicting sort of growth and change in tumor numbers in the sensitive and resistant cells in those mixed tumors, it looked like there was a huge mismatch between what we would expect based on the growth rates of monotypic tumors and what we observe in these mixed tumors. So there were way more sensitive cells that we would otherwise expect and way fewer resistant cells, which suggested there is some sort of biological interaction happening here. At that point, and if we kind of introduce this interaction term in the models, now we fix the discrepancy. But what is this interaction? And at that point, we already had a good clue that it might be this interaction mediated by stroma. And so when we stained for the location of this resistant and sensitive cells, I think the picture really kind of jumps out where outside of the stromal niches, the resistant cells are taking over. They are undergoing essentially competitive release, but closer to stromal region, we're seeing that enough of the treatment naive cells are capable of surviving. And they're not just surviving, they're changing, they are acquiring resistance through different trajectories. And again, Tatiana could capture these differences with her spatial analysis models. And so what we think is going on is a very interesting phenomena of essentially ecologically mediated positive interaction and ecological rescue that precedes evolutionary rescue, where in normal scenario, the naive cells are partially protected by the stromal niches, but still if the therapy shrinks the tumor, you will getting fewer and fewer of these niches that can support the naive cells. The resistant cells, however, they don't care about the stromal proximity, they expand, and by expanding, they're limiting the loss of the stromal niches, and then as they mediate the outgrowth of the tumor, we're adding new and new niches that can support not only those resistant cells who don't really need them, but also the sensitive cells who can exploit it. And so it leads to kind of very interesting and unusual dynamics that would be completely missed if you don't if you don't consider the stroma. And again, like since due to time considerations, I won't spend any time, but we have a preprint about a different type of the effective stroma where the resistance might be mediated without any directly impacting sensitivity of tumor cells, but by enhancing the baseline proliferation, where the small differences in proliferation close to stroma can facilitate the recovery of tumor cells during the cycles of chemotherapy. And Tatiana is going to talk about this story tomorrow. So a few key conclusions from this talk, and from our studies is, first, the main message that, and we still have discussions with Joel Brown and some others, but I think that that cancer cannot be reduced just to ecoevolutionary process. You cannot understand it without considering ecology and evolution, but you need to, in some cases, take a different considerations and think from the perspective of messed up differentiation and tissue homeostasis. To understand cancers, we need to consider tissue dynamics. We need to consider turnover rates, proliferation, death. We have to think about it in space and think not only about what's happening intrinsically in tumor cells, but what also might be happening in the environment. We're seeing that resistance is acquired through a dynamic and multifactorial process. So if you try to reduce it to a pre-existing resistance, maybe there are cases where it worked, but there's also a danger of misunderstanding or jumping into clinical trials based on wrong assumptions and ending up hurting the patients. And the last point, we need to account for the tumor-stroma interactions to make sense of the evolutionary dynamics triggered by the tumor's response to therapies. And with that, I would like to thank the members of my lab and the extended network of collaborators, which makes it fun, and the fourth floor and Sandy and other IMO members, which makes it a really kind of unique and cool environment for working and training. So if you're looking for postdoc opportunities, definitely look into IMO faculty members. And I'm thankful for the funding sources, obviously, to keep us being able to do these studies. Thank you, and I'm going to take some questions. Looks like there are 5 minutes for questions. I tried to be quick, so it's worked out.
Thank you, Andriy. Regarding the EMT, I have seen that it's frequently related to individual cell migration. And I was wondering if you observed in your co-cultures any visual signs of individual cell migration.
Yeah, so in the cultures, that's true. The EMT, well, the cells are more migratory. And one interesting thing, and which kind of might be useful, important to consider with modeling, what EMT often does, it leads to apparently faster growth rates, just because the cells, if you have cells that cannot migrate, they get crowded out by themselves, so you can sometimes get the clumps of the cells. If you induce EMT, the cells can crawl to the new space and explore. So basically their effective proliferation rate might be, so you might have cells with similar potential proliferation rates, but the migration enables cells to explore space more effectively. But definitely you do see cases where you see individual cell migration. In vivo, things can be quite complicated because there's also collective migration happening, but we don't have the tools to look at that, and we haven't done any of that. Thanks.
Thanks a lot, Andriy. So much to unravel, but I'll start with one question. So for the tumor initiation things, you made a very interesting point that I hadn't thought about, and that the capacity to actually grow in 3D might be crucial, right? So if we have monolayer tissue in epithelial, so where we actually have the stem cells at the bottom, and then we start to differentiate as things go up, people have argued that actually the ability to stay on the bottom layer is crucial for precancerous inflation of that cone. So in the context of field canceration. So that strikes me as, oh, actually that's weird, because so before you actually start to grow in 3D to expand, you need to stay 2D in order to proliferate and compete with your neighbors. But then maybe it's important to suddenly switch to 3D modes in order to then proliferate and actually form a cancer. Have you thought about this, I guess, like by modality?
Yeah, that's a kind of very good point. It's very important point. So one thing, again, like I think what sort of cancer, during the cancer progression, there seems to be a, like the whole thing of stemness is a little bit of a minefield. I think it's better to think about a sort of epigenetic plasticity or developmental plasticity, but there seems to be a progressive acquisition of promiscuity of what cell can consider the niche. And there's a kind of interesting observation that like contacting with basal membrane produced by stroma provides the niche for breast cancers, but and for many other epithelial cancers, but it looks like getting in 3D, and there's like Mehdi Demaghi has a cool paper about it. It looks like, and we're seeing the same thing during the development of resistance. So it looks like the cells that get outside of the niche, sometimes they basically secrete their basal membrane and they provide essentially the niche for themselves and for the neighbor. So it can lead to a very weird and interesting dynamics, but that would, I guess, like would be context specific, and that would require kind of careful examination before coming up with figuring out what the best way to model it.
Yeah. Maybe like a stupid naive physicist perspective, do you think it could be buckling actually? So that you have 2D growth and at some point you start actually to buckle, and that's the transition to.
Yeah, so it looks like, again, it looks like it might be happening in like in colon crypts. Oftentimes there's like hyperplasia that seems like buckling. It doesn't seem, again, like happening too much in breast. And it looks like they also have a kind of effective tumor suppression mechanism where the cells, like if it gets kicked out, where the potentially malignant cells can get kicked out of the layer, and then most cases they die, and usually they kicked up in in the duct, they get kicked up in the lumen, and well, you shit it over, it's no problem. In the breast, it can remain in the duct, and if it doesn't die, it could create a problem. If it figure out how to kind of survive on its own by producing its own matrix, it could provide a problem. And at least in the DCIS, in many cases, it looks like there's still some normal cells that are staying there, and there are now have this kind of more malignant cells that kind of poison the environment. But there's definitely cases where the buckling effect is happening.
Super, thank you.
Really cool stuff. So I don't know if this is relevant for long, but apart from the fibroblasts, right, inside the stroma, you have fat cells. And do you see any effect of how many fat cells or recruitment of fat cells around like these more resistant tumor spaces?
Yeah, so good, very good question. The thing is, well, the fat cells is the same kind of lineage as fibroblasts. So adipocytes can be differentiated into fibroblasts, mesenchymal stem cells can become fat cells. We are seeing evidence of like being close to fat accelerating tumor growth, and there's kind of good studies that describe the sort of metabolic crosstalk. We're also seeing fat cells inside of the tumor tissue. But at this point, we haven't started trying to figure out the details. Obviously, the fibroblasts are not homogeneous. There are different types of fibroblasts. They're adipose cells, and they all kind of have different flavors of the crosstalk. So we haven't started looking at these details, but these details matter. And so there's definitely something to it.
Okay, thanks, Andrea. You'll introduce the next speakers, but I want a question as well. So one of the things that you touched on was about the normal architecture of the organ. And of course, in lung, a lot of the times the cancers are in ducts. And that duct architecture is in many ways separating the stromal environment from the non-stromal environment. So there's this whole process of the tumor potentially forming and evolving in an enclosed environment before it breaches it, right? So when it already breaks out, it's already been through quite an evolutionary crucible in some sense. And so I guess my point was that in lung, there's different, you get tumors in, is it squamous versus adeno or something like that? One's in the duct and one isn't. And so I just wondered if when you emerge from that, is your stromal interaction different versus if you're already like embedded in the stroma, you just breach it right away?
Yeah, so I'm not sure if I have a good answer to your question. I would like, it makes sense that it would matter. And oftentimes the sort of histology is reflection of, again, sort of different caricatures of normal things, but also reflective in many cases cell of origin, where they can make sort of more flat sheets versus kind of different structures. But even like in those examples of like xenograft tumors, one can say that sort of these kind of layers of stroma that surround the tumor cells, that there is a resemblance of the kind of ductal structure where the cells that are close to the stroma might be kind of more similar to the cells that normally reside on the basal membrane, and these cells have higher stemness potential, and it impacts a lot of things, metabolism, gene expression, etc. So there is kind of a remnant of this structure. But the other interesting thing is that as tumors progress, typically you are kind of losing more and more of the resemblance of normal tissue. So they become kind of less and less dependent on the normal interactions and more autonomous. So that's why I don't get this, so that sort of the cancer is an speciation and acquisition of things. That seems to be a kind of a process that can be quite protracted, and it's difficult to say where is the clear kind of watershed moment is.
Okay, thanks, Andriy.